So we are learning in lecture that because we're a diploid organism, we receive 2 copies of each chromosome. We can receive a dominant version of an allele or a recessive version of an allele, if we're only talking about one or the other. So that means if, let's just make up this scenario here where I used the letter A just to kind of as a placeholder. Let's say that whatever this gene is, it only comes in 2 forms, either dominant or recessive. From either parent, depending on what their genotype is, you could receive 2 dominant copies, one from one parent, one from the other. Both parents could give you a recessive allele, or one parent could give you a dominant allele, and one parent could give you a recessive allele. Those are the 3 conditions when you're dealing with dominant versus recessive. We call these homozygous dominant, homozygous recessive, and this one down here, heterozygous. There's no such thing as heterozygous dominant. That doesn't make any sense. It's just heterozygous. And we've kind of talked about in lecture how if we have this one, our only option, if this is the gene being transcribed and translated, we'd make the dominant protein. With homozygous recessive, obviously we'd make the recessive protein. But down here, it gets a little trickier. If we have a copy of the dominant and a copy of the recessive, and we're dealing with dominant versus recessive, not a blend, we only make the dominant protein. This one is turned off, like it doesn't even exist. It's not transcribed, not translated. And so we say it's masked or turned off. We only make the dominant copy. That's kind of the basics. We learned that a little bit in lecture. With blood, it's a little trickier. Because blood comes in multiple forms, and it's what we call co-dominant, we don't just write it with one letter. When we're writing the genotype for blood, you write a capital I and then the dominant allele. So let's talk about the different blood types, and then we'll go back to those genotypes. You've probably heard of your blood type before, right? Even if you don't know what you are, right? You've heard type A, B, O, positive. If you are type A, what that means is on the outside of your red blood cells, you have special proteins. If you think back to Unit 1, we learned how the outside of all your cells have those glycoproteins that allow your body to recognize you as being you. We call it the MHC complex, or we also said the sugar fingerprint. We talked about how if you got an organ transplant, even if it's the same blood type, your body still may fight it off because it doesn't see it as being you. Do you kind of remember that conversation? Your body, if you are type A, these red blood cells have all that special protein on the outside. They don't really look like the letter A. I just drew that to make it easier. But you could say that you have the A protein or A antigens. That means your body recognizes that as being you. But your body does something weird. To protect you, it makes a bunch of antibodies all the time. Even when you're not being exposed to things, it's just constantly trying to make up new antibodies just in case you come across something. With blood, it makes an antibody against the other condition. So if you're type A, out in your blood serum, you make anti-B or antibody B. If you're type B, same story, you make B proteins around your cells. But out in your blood serum, you produce anti-B. Let me turn this light off so it's a little easier. If we skip over to this one, if you're type O, it's really type 0. You don't have anything on your red blood cell. Neither of those antigens, neither of those proteins. So your body produces antibodies against the other conditions. So if you're type O, you make antibodies against A and against B. There's one more type of blood, though. Type AB. If you're type AB, you make both the A and the B proteins. And I gave away the answer. I was going to quiz you, but I already wrote it on there. That means your body recognizes both conditions. So you don't make either antibodies. I'll zoom out here. And all this is written on the board as well. So if you miss something, it's behind me or behind the screen in the other room. It is. And so this O we call the universal donor, because when you give blood, they just give the blood cells. You can separate it from the antibody A, antibody B. So if you're just giving this red blood cell with no proteins, it can be given to anybody. So type O folks can give it out to anybody. That's why if you're a donor, if you've given blood before, they call you a lot if you're type O because they want you to give it because it could save so many more lives than the others.
I thought O negative was the true one.
It is.
Positive can't
Correct.
do all of that.
We've not gotten to positive negative yet. That's the next step. But you are absolutely correct. O negative is the universal donor. AB, because they don't have any antibodies, they can receive blood from anybody. However, because their blood has A and B on it, they can only give to AB people. Because let's go back to type A. I skipped a step. If we were to give A to someone with type B blood, this antibody would glob onto that. It's called agglutination. It would clot, essentially, inside of your arteries and veins, and that is not good. Clot a lot faster than your body could deal with it.
So type B can't receive type A.
Correct. And type A can't receive type B. Type O can only receive type O because they have antibodies for both A and B. So they'd interact with this, it would interact with this, and it would interact with that. And so while folks with O blood are kind of the universal donor, they're at a disadvantage. And so if we're ever in a post-apocalyptic Walking Dead style situation where you need to get a blood transfusion, it's good to know your friend's blood types. I'm type O positive, just so you know, in case we see. So every semester I should be making a list of these things, like, just so we know. But you brought up a good point. Before I get into, well, let's go up here. If we're looking at the genotypes, the actual genes, to be type A, you could be dominant for both. So you received a dominant allele from one parent, a dominant from the other. We write that as the letter I with a capital A to the right. That just tells us we're dealing with a co-dominant blood type. Or you could be heterozygous, dominant A, IA, and a lowercase I. That's the recessive condition, is lowercase I. Whoops, I didn't point to the right spot. There we go. If you are type B, you could be homozygous dominant, capital B, or heterozygous, capital B, lowercase I. I said the word co-dominant. Look here at type AB. If you received an A from one parent and a B from the other parent, at the end of chapter 11, we'll see the vocab word co-dominance. Neither of these alleles mask the other. They're just both expressed. You just make both proteins. So that's why your blood has both of those proteins on the outside. Type O, however, homozygous recessive. You don't make either protein. But you brought up a very good point. There's one more condition. You've probably heard if you're O positive or A negative or something, there is another protein. When they did the primary research into this protein, they used the rhesus monkey. It's a small New World monkey. And so they call it the rhesus factor or RH factor. And when we write it in Mendelian genetics, we just use the capital letter D. I don't know why. I've not looked that up. It's just what is done. If you are positive, that means you have this RH factor. You have this protein. So you could be homozygous dominant or heterozygous and still make this rhesus protein. If you are negative, you don't make it. So you're homozygous recessive. you develop antibodies against the protein if you are negative. So you're exactly right. That's why O negative are the universal donor, because they don't have the rhesus protein and they don't have the A protein or the B protein. So they can just give that O negative blood to absolutely anybody.
What does rhesus really mean? Like, maybe you said it, but I don't like.
It's RHESUS. It's just the name of a monkey. It's called the rhesus monkey. They have them at the zoo in St. Louis. And because they did the primary research on those monkeys, they just named it the rhesus factor. And so now we just call it the RH factor. If that the video does I have it written on the board behind me, but if you needed more information, it's also in that makeup video if you wanted to watch that. If you are negative and you are pregnant with a positive child, we learned that the blood does not mix during pregnancy, or it shouldn't. The placenta and the fetal blood is kept at a barrier. However, as we saw with Melinda, even though she had a pretty simple, in quotations, simple birth is kind of traumatic. And so blood does get mixed during birth. If you are negative and your baby is positive, after birth, you've probably experienced some of that positive blood in your system. So you make antibodies against it, because you always make antibodies against the opposite condition. If that's the case, if you're pregnant again with a positive baby, your body could fight it off. And so they will give you special shots called RH shots or RhoGAM shots to make sure that you don't fight off your positive child. That's why, if you've ever experienced that or heard of that, it's because your body, once it has experienced that positive, makes the antibodies against it, because that's just your body's function. It just likes to make antibodies to help protect you. And sometimes it's good, sometimes it goes a little too crazy. And you should find 5 white trays. We'll line them up all the same way. That was a little dirty. Oh, I didn't clean these out very well. Oops. You should also have 3, oops, turn my lamp off. What do those makeup tutorials do with the hand, like to make sure, there we go. I can't do it. 3 toothpicks, a yellow, a white, and a blue. We are not throwing these away. They're plastic. We can reuse them every time. You may have to get a paper towel and wipe them off today, but we'll keep them. Some people already have a paper towel. There you go. You should also have 5 blood samples. June, Andrea, sample 1, sample 2, and sample 3. So that you don't get confused and accidentally get things mixed up, you should line your blood types up in front of a different tray. That lets you keep things straight. We're learning that part of good science is a good experimental design with nice quality data. So if you separate it like that, you won't get confused. You'll also have 3 different antibodies or antiserums. Antibody A, ignore if yours has mildew. I don't know why they're growing mildew and mold. Antibody A, antibody B, a yellow one, and anti-D, a clear one. That's the RH factor. Remember we just used D for RH. I'll look up why one day. I've not looked it up yet. I think it's because we have actually 20 to 30 different blood types, and RH is just one of them. But anyway. We have a mystery. We're pretending that we're working in a clinic. We're doing types of sampling, blood testing. June has a daughter, Andrea, and she's pursuing child support. And we're trying to figure out who the potential father could be out of 3 different samples. Sample 1, sample 2, sample 3. Somebody asked last night why they don't have names. If you ever do work in one of these clinics, to reduce bias and reduce gossip and that type of stuff and HIPAA violations, they often just label things sample, because if you're the technician, you have really no reason to know the names, you're just trying to figure out what the blood type is. So they label them sample 1, 2, 3. If you look at these trays, like you can't see it on my projector, but there will be 3 wells, and each well is labeled A, RH, or B. A is at the top, B is at the bottom, and RH is over here. We're going to figure out what blood types we're dealing with. If an antibody touches that blood type, it's going to clot. So if I have anti-A and it touches a type A blood, it's going to clot. Now our clots are going to look different. Some will be really chunky, some will just be a little liquidy with little dots in it. So I'm going to do the first one together with you, because sometimes these blood types, they need a second. So what we're going to do is take our antibody A, the blue one, and we'll put it 1 drop, so all you need is 1 drop, in the A well. I like to set all of mine up at the same time. You can do it one at a time, or just go through and put a drop in each of the A wells. Since I have it open, I just go through the row there. Once you do that, take anti-serum B and put a drop in the bottom B well. And then you probably guessed, you'll then take anti-D and put it in the RH well. Just 1 drop, 1 drop. Our toothpicks are color-coded. In between samples, we'll want to wipe them off so that we don't cross-contaminate. But they labeled the blue toothpick for anti-A, the yellow for anti-B, and the white one for anti-D. That's so we don't accidentally cross-contaminate and mix up our results. Let's start with June. Let's figure out June's blood type. She's the mom, she's the first one. So you may need to shake it up a little bit. Open it up away from you. If it splashes on your clothes, it is stain. And then just take 1 drop and put it in the A. All it takes is 1 drop. And then take your blue toothpick and stir. Am I on camera? Yeah, there we go. Nothing is really happening. So that means that serum is not interacting with that blood type. So she must not be type A. So down at the bottom of your worksheet, you have a chart with the different names and the blood types on the left. For June, we can put no, or an X, or a 0 where it says type A, or blood A. I just use no. Let's move to the B. Put 1 drop of June's blood in the B well, and then take your yellow toothpick and stir. Can you notice the difference? If you don't, keep stirring. Do you see those tiny red dots that are forming? That's clotting, or agglutinating is the word, agglutination. And so for B, we'd have to say yes, because it looks like June has type B blood. Now to figure out if she's positive or not, we do the same exact thing. Just take the drop of blood and put it on the positive, or on the RH, I'm sorry. Take your white toothpick and stir, and not a lot's happening. Some of these need stirred for about 15 seconds, but nothing is happening. So we can say no on type RH. The RH serum sometimes takes longer. And so you might have to let it sit for a second and then stir it if it is going to clot. Sometimes these samples are old and we just have chunks of old blood that look like clots but aren't really. So what I'm going to do is, after we've gone through and you go through each of them just like that, we'll talk about it and I'll give you the right answers.